Hello and welcome. Welcome back to another of OpenEye's webinars. Today, I am delighted to have with us our head of science, Dr. David LeBard. David's background is in theoretical and computational chemistry and biology. In his 8 years or so at OpenEye, he's worked on membrane permeability prediction, cryptic pocket prediction, and pocket ligandability modeling. And it's on these latter two subjects, cryptic pockets, pocket ligandability, that he's going to talk to us about today. So, without further ado, David, please take it away.
Thanks, Paul. And thank you everyone for joining me today. I'm really excited to talk to you about some work that we've been doing over the years here at OpenEye, where we're trying to develop a method to help drug the undruggable. And in that, we're trying to develop a set of methodologies to help both detect cryptic pockets and also rank them based on their ligandability. So there's a few questions that we're asking in order to kind of answer the question of, whether or not we can make a solution for detecting and ranking cryptic pockets. And those questions start out with, where might a ligand bind? So given a protein molecule, the most simplest question is, on this protein, where are the possible sites where a ligand could potentially bind? And then you refine the question down to where a drug-like molecule will typically bind. And this is really where you want to start the structure-based drug design campaign, using a pocket that a molecule will actually bind to. And finally, what happens if a molecule actually binds there is some biological effect. And if we want to go ahead and start in the beginning, what we have to have a crystal structure for any type of structure-based drug design. And normally we start with X-ray crystal structure, but this can be really limiting. And I think that you can be trapped by the structure. And the reason why is because proteins actually move. And I'd like to kind of harken back to what Galileo said when he was being hauled off for heresy, when he supported Copernicus's idea of heliocentricity, which says that the sun actually is the center of the center of our solar system, and the Earth revolves around it, not the other way around. But as he was being hauled off by the Catholic police, he said, and yet it moves. And I think we need to kind of, take this statement into the stance of structure-based drug design. We can't just be trapped by a crystal structure. These proteins move around, and we have to move them to see what they do. And you can do that by using molecular dynamics, and it might take quite a lot of MD. Or perhaps you have another way of doing it, and maybe you could do slightly less MD and open pockets using something like a chemical probe. And we have ideas about how to do that. Now, once you've done some MD, then you have to observe pockets, but that only tells you about where might a ligand bind. And now you have to actually rank the pockets to get to the point where you can tell someone that this is a good pocket for starting your structure-based drug design. So let's go ahead and try to answer the first question. How can we come up with a solution where might a ligand bind? So in order to do that, we've been thinking really hard about the types of motions that are associated with cryptic pocket formation. Some of them are relatively simple, like side chain motion, which happens on the order of nanoseconds, where you have a simple side chain that needs to move out of the way in order for a pocket to open. Or you might have loop motion, where you have an entire loop that needs to get out of the way for a pocket to open. Or perhaps the reverse process, where a loop has to come back on top of a pocket and form a lid. Alternatively, there's even longer timescale motion, like secondary structure motion, where you have to move an entire secondary structure element out of the way or bring it on top like a lid. But this also is, a little bit more time consuming, and already microseconds to seconds is outside of the scope of typical simulations. There's even more complicated scenarios where you have secondary structure that needs to change, where you go from some order to disorder state, or maybe the reverse process, or maybe even a really long timescale motion like interdomain motion, where you have two parts of a protein that may be tethered or not, and they either have to come apart to expose a pocket, or maybe they're already apart and have to have the reverse process to come together to form that pocket. And finally, there's another type of pocket that some people do not consider to be actual cryptic pockets, but they are pockets nevertheless that still need to be detected and ranked. And these are pre-existing pockets inside your protein. And I think of these like little holes inside of Swiss cheese. And these are the kinds of motion and pockets that we want to be able to detect. So in order to do this, we need to go way beyond the timescale of what molecular dynamics can do. So how can we do this without adding any bias to the system? Well, what we're going to do is use a method called weighted ensemble to provide the kind of speed up that we need without this kind of bias. So the way I'll explain this is through the process of protein-protein binding. And this example was taken from my collaborator Lillian Chong at University of Pittsburgh. And here what she was doing, she was taking the proteins Barnase and Barstar and binding them, starting from the unbound state at the top of the screen to the bound state at the bottom of the screen. And the methodology requires a set of weights where you rigorously track the weight of a given trajectory that follows the process of binding in this particular case. And you have to have some measure of how much progress you're making. And so we call this measure a progress coordinate. In this particular case of protein-protein binding, there are two coordinates that were important. There was the binding RMSD, which is the RMSD of the interface of it being in the bound state. And then there are also the minimum separation distance between the two proteins. And the way this method works, you start out with an ensemble of protein structures that are started in the unbound state in the top right-hand corner, and you want to get in the bound state in the bottom left-hand corner. And so you start a number of trajectories that are integrated stochastically, so they have a little bit of noise in them. And because of that, they decorrelate a little bit, and you run really short trajectories. And then you ask the question, are you making it to a new point in this particular progress coordinate space? And if you are, you're like the green trajectories, and then you get replicated, and your weights of that individual trajectory get split. But if you're not making any progress, like the green or the purple trajectories, then we take those trajectories and then we collapse them down to one, and we stop doing the one that's not making any progress. And we actually save computing power and then follow the winners such that they go from the unbound state to the bound state. But if you want to do this for something like cryptic pocket exploration, how are you going to do this when you don't actually have a coordinate system that tells you how to open a pocket at all? So the way we decided to do this at OpenEye is by using the intrinsic normal modes within a protein. And these will likely explore protein motions that are inherent to the molecular structure. So the way we do this, we take our protein and we make a C-alpha based network model from that. And then we take that network model and we calculate the Hessian and we diagonalize it to get a set of normal modes, which are expressed as a set of eigenvalues, which is given by lambda here, or eigenvectors, which are pointing to the direction of the motion that you're interested in. And there's this really interesting relationship between the amplitude of the motion on the left-hand side, the squared amplitude in this case, and the energy of the motion, which is on the right-hand side, also relating to the eigenvalue, which is lambda, and the frequency of that mode, which is omega. And so the way you can think about this is that if you fix the energy, then you get the largest amplitude from the lowest frequency motion, right? And another way of thinking about that is that if you fix the amplitude, the lowest frequency motion, the one that gives you the kind of motion that you would like to think a protein would open up a pocket, actually requires the least amount of energy. And this is what we're going to rely on to open up our pockets. Normally, when people think about these normal modes, they talk about them in terms of their so-called fractional variance, which is shown here, but it's related effectively to the frequency of the motion. And you basically take all of the modes that you calculate, and then you stack them up based on the fractional variance. So the lowest modes have the largest fractional variance, which means they have the slowest, most largest scale motion, and they decay as the mode number increases. However, what we have realized is that some of these low frequency modes that give rise to the largest motion may not be appropriate for opening up pockets. And for that, we've used a different metric called collectivity, where collectivity measures how much of the protein is actually involved in the given mode of motion of a given mode. And if we take the collectivity and see how it's lined up with respect to the ranking based on the fractional variance, what you see is that some of the higher mode numbers actually have larger collectivity. Now, what does this actually look like? Well, if we look at the eigenvectors that are projected back onto the proteins themselves, the modes with the most collectivity, modes 14 and 15, shown at the center of the screen, you can see that they have eigenvectors that are effectively isotropically distributed across the protein surface, whereas mode 12, which is really focusing motion on a particular region, doesn't have that. And you can see that the collectivity drops quite a bit. So what we're going to use for our exploration of protein conformations is the most collective of the lowest frequency modes. So how can we use this to actually get our protein to move around in a smart way? We're going to combine this weighted ensemble strategy that requires a progress coordinate and our normal modes, which will be our progress coordinate, and use that to explore protein conformational states. So what we'll do first is we'll run an initial analysis to get the normal modes, that's the eigenvalues and eigenvectors. And then we're going to run a short MD simulation for an ensemble of trajectories. And we're going to recalculate for each one of those our same modes that we originally calculated. And then we're going to project those modes back onto the original modes to see how far along a given direction we've actually moved. And then we're going to cycle along this again and again and again until we've done appropriate amount of sampling. And in this way, we're allowing the protein to explore conformational states in a really fast and efficient way. But of course, there's a catch. Now, the catch is, what if this isn't actually how you find a cryptic pocket, right? So what we're doing here is we're pushing the protein around and having it explore conformational states, but maybe it requires some type of other effect. And in fact, it might be energetically unlikely to open up some pockets because it might require some chemical probe. Now, what do I mean by this? I mean that in opening cryptic pockets, there's really two axes that I tend to think about. One is a conformational selection axis, where the protein is interacting with a probe molecule, which is water, at a very high concentration. So you have a really greasy thing inside a really polar system. So it moves around naturally based upon this. But there's another axis, which is the induced-fit mechanism axis, which requires a chemical probe to be there in order for, the energetics to be more likely. And it's possible the pocket opening happens somehow as a linear combination between these two. But what are we going to do for this induced fit mechanism? What goes here on this x-axis to be our computational molecular probe? Well, could we use something simple like Xenon to perhaps open pockets? That's the question that we have. We think it's suitable based on knowledge that we have about this particular atom, not even a molecule. But there's really two ways encrypted pockets open. I just explained this on the last slide, but I'm going to go a little bit more in detail here. So one, you have conformational selection mechanism. This means that the protein opens up by interacting with water. And what you're seeing here are residues that are shown in orange. They're changing their solvent exposure based on the interactions of the protein with the water itself. Now, the induced fit mechanism is slightly different. Here, you see orange residues that appear not because they're more solvent exposed, but rather because they're binding probe molecules. Now, these can be experimentally verified, both of these mechanisms, and they have an observable, which is the ligand concentration. So if you're thinking about a conformational selection mechanism, there is no ligand to be present that's necessary to be present. And so you have no ligand dependence. However, if you have a conformationally or an induced fit mechanism that your pocket requires to open, there is a strong ligand dependence. So if you add more ligands to it, it's more likely you're going to find a suitable protein nearby, and therefore you're going to have a strong ligand dependence on the pocket opening. So that's the way we see these two mechanisms working. So the idea, could we really use Xenon as a reasonable chemical probe? We think we can, and it's based on some observations. So number one, we know that Xenon acts as a general anesthetic. So what does this mean at the molecular scale? Well, if you have an ion channel like the GABA A receptor and you add Xenon to it, it will cause the receptor to actually close, and you can see macroscopic changes happening in the current that goes across the membrane. So it stops the flows of ions that affects the central nervous system. Another aspect that we think is important is that xenon actually acts as a neuroprotectant. And what do I mean by that? I mean that it promotes neurogenesis and it prevents apoptosis or programmed cell death. Because of this, we know it has to have some interaction with proteins that causes a dramatic shift in the conformational equilibria. And because of these two taken together, we think that xenon could potentially stimulate pockets opening through an induced fit mechanism. So we have two methods now for detecting cryptic pockets at OpenEye after we've applied our protein sampling method. So this first method is called exposon analysis. This was developed in the lab of Greg Bowman, and it uses correlated changes in solvent exposure, like I explained before, in a way to define what residues are involved in pocket opening. We were highly inspired by this method and developed our own method called dynamic probe binding analysis, where if you run a simulation in the presence of xenon using our protein sampling protocol, we have a analysis method that can look at correlated changes in binding propensity of xenon to the protein. And these are two alternate methods, one exposon method that allows us to look at that conformational selection axis, one dynamic probe binding analysis, which lets us look at the induced fit mechanism axis. So we have a case study that we've been working on over the last few years called KRAS that many of you likely know. I'm just going to give you a little bit of background on KRAS in case you aren't familiar. So KRAS is a molecular switch. It's active when GTP is bound and inactive when GDP is bound. However, there's some oncogenic mutations, primarily around glycine 12, that keep it on the completely on the on state for its entire lifetime. And this has many effects. Most notably, it causes tumors to grow. So what happened is KRAS was discovered in the early 1960s, the gene for KRAS. It wasn't until the 1980s that it was found to be a very important target for cancer therapies. But it took about 30 years before people were able to find the switch 2 pocket in KRAS, which was discovered by some work from Kevin Shokat's lab at UCSF, where he was doing some covalent fragment screening and was able to find that this switch 2 pocket is potentially suitable for therapeutic design. And 10 years later, we now have two FDA-approved drugs, sotorasib and adagrasib, that actually bind to that site, and several others that are in the clinic. But if you think about the story of KRAS, the rate limiting step for developing these therapies, which this protein is involved in over 10% of all human cancers, so it's really important. But the rate limiting step wasn't finding an actual, drug-like molecule, it was finding the pocket. And so what we'd like to do is remove that 30-year wait and help you find these kind of pockets with the push of a button. So if you look at KRAS, on the left-hand side, we see the apo structure without a ligand bound. On the right-hand side, we see the holo structure with the ligand bound. And the main difference you see is that there had to be some dynamics for this pocket to open. And the time scale is, potentially quite long, upwards of seconds, which is far beyond what typical brute force simulations can do. And the length scale is quite long. You have to really move loops around. You have some interdomain motion and other things. And so how are we going to be able to do this? Well, we're going to use our normal mode driven weighted ensemble method to hopefully open up these pockets. And then we're going to take our methods that we talked about before, this exposon method and dynamic probe binding, and we're going to analyze this protein to see if we're able to detect those pockets. So this is the results of that study, which you can read. It was published in JSIM late last fall. And here I'm showing you just the results for the switch 2 pocket that was discovered by both the exposon method and the dynamic probe binding method. And you can see that they discovered the residues that quite nicely line what is accounts for the switch 2 site, which is where this Mirati 1133 compound is binding. But if we take a deeper look at our simulations, one thing that we thought was interesting is that in all of the holo simulations where our pocket is open, we find that tyrosine 71 is solvent exposed. However, in the apo structure, we find that this tyrosine 71 is actually occluding the pocket. We suspect that this tyrosine 71 in that particular case may act as a gatekeeper for this particular pocket opening. But there are other sites that we found on KRAS, and at the time, we didn't have a ligandability model that I'll tell you about at the end of this talk. And we found lots of sites, and we were concerned that some of them may be actual false positives. So we had to go through the literature and determine whether or not these sites were real or not. And so we looked at them, and site number one was actually the site where all of the molecules bind at the switch 2 pocket. And in fact, where Xenon tends to aggregate is at the most potent portion of where molecules tend to aggregate in the switch 2 pocket. So that was also pretty interesting. If we look at the combination of sites 1, 2, and 3, this is actually where some work for a peptide inhibitor has been going. If we look here at site 4, we find that this is a site that has not been talked about that much. This is the switch 1-2 pocket, and this is on the other side of switch 2. And we thought that this was a false positive, but as it turns out, many compounds, and in fact, one of the analogs for Mirati 1133 actually bound there as well. There are other sites here that we don't know about, and in order to classify them, we need a model or something that can tell us that these pockets are actually so-called ligandable or not. So what are we going to do for that? Well, first, we're going to go ahead and open up our validation set, not just look at KRAS, but also look at different types of pockets, like I mentioned in the beginning of the talk, that open up under either sidechain motion or loop motion or secondary structure motion, secondary structure changes, or even deeply buried pockets. And if your favorite protein isn't mentioned here, please reach out to us. We'd be happy to take it as a challenge. A few of these were challenges from our friends in the field. But just now asking the question, are any of these potentially ligandable? That's answering the first question that I brought up. Where might a ligand bind? And if we look at this data set, what we see is that using our cryptic pocket detection method, we're able to detect pockets on 91% of the proteins that we tried. And the failure cases that we came from were one, secondary structure changes, where you have to go from an ordered state to a disordered state to another ordered state. Naively, I would like to blame the force field people for this, because they typically like to make force fields that'll maintain secondary structure, but I can't necessarily say that. I think probably we need to improve our method. But also, we have another failure here in the deep pocket finding, where you have a pre-occluded, you have a pocket that's already inside a protein. Can we detect it? There's one particular case where we weren't able to. We suspect this might be due to our low concentration of Xenon, and we're still looking into that today. But now we want to refine our method a little bit further and ask, where does a drug-like molecule bind? Not just where might it bind? Is this thing even ligandable? But rather, where should I start my drug design, my structure-based drug design campaign? So we need a method that can actually separate good pockets from bad pockets. And so I'm going to tell you about a model that we've been developing here at OpenEye to give you a sense that a pocket is more than just finding a groove on the surface of a protein. So I'm going to give you a little bit of background on ligandability assessment methods, just to go into what our method looks like. So a lot of this work has been progressing over the last few years. It started out in about 2007 with the PLB index, where they trained on a mere 41 structures, and they asked the question, what is the propensity of a given amino acid to be inside a binding pocket? And so they look at a pocket and they say, well, proline is really unlikely to be there, tryptophan's really unlikely to be there, and based on the residue composition, I give it this particular score. Now, about 10 years later, P2 rank came around. They upped the training set by about half an order of magnitude to about 250 structures. And they used random forest model. Instead of looking at the surface residues, they look at surface points. And each point on the surface of the pocket gets a little score. And based on the sum of the score of the surface points, that's how you rank a pocket. Now, there's this really cool method that came out last year from Patush Tawari's lab called KRAS. And this is a graph-based attention neural network. And this uses surface atoms as the model. And it was trained on about 17,000 structures and 26,000 binding sites. And I'd like to argue that there's still some issues with these, not because the models are necessarily bad, but we think that the training data was not necessarily appropriate for making the ligandability model. So the questions we have are, do the existing models use truly non-redundant data sets? And how they split the data sets up were based on two criteria. One was protein sequence identity, and another was the structural similarity to a given protein family. So these have some issues with them, and I'd like to point them out now. And one is that protein sequence in and of itself, I don't think is sufficient because it doesn't reflect 3D structural similarity. And what do I mean by that? I mean that the same sequence can adopt two completely different conformations. I think one classic example is calmodulin that doesn't look anything like calmodulin when it has a calcium bound to when it doesn't. But then you also have, completely different sequences that actually can lead to the same fold that can lead to the same enzymatic activity. Now, there's also other issues if you look at protein families, because many protein families have diverse binding sites, but they also bind the same types of molecules, and so you might be overfitting on some of the same chemistry, and we think that might be what is happening with some of the models that are out today. So if you look at the SCP PDB bind dataset, which is what GRASP trained on, it's unfortunate, but about 50% of all of the training data comes from only 3% of the ligands. And it's because they didn't really look into the ligand chemistry when they were trying to do their splitting. And if you ask the question, what kind of ligands are those? Well, they're mostly nucleotides or nucleotide derivatives. And we think that that is causing an issue where you're not able to make really strong predictions of pockets that don't necessarily bind nucleotides. I think this is a good example. So if you take the GRASP model and you apply it to our favorite system, KRAS, and you ask the question, what of the three pockets that we know about is the most ligandable? And it will completely identify the GDP pocket as a really ligandable binding site, but will ignore the FDA approved site and the switch 2 site, the switch 2-3 site. So this is an issue if you want to use a model like this in prospective work. How are you actually going to identify sites that aren't just nucleotide binding sites? To answer that, we tried to make our own data set that removes some of these issues. So we tried to use pocket similarity for preparing our data set and use a smart splitting based on this in order to not have any type of leakage from our tests to train. So this is how it works. We rely on a method that we developed at OpenEye many years ago called SiteHopper. And SiteHopper is effectively a ROX overlay of two binding pockets. So what you're seeing here on the left-hand side is a reference pocket in green, and this has a set of shape and color features that describe effectively what the shape and electrostatics of the pocket looks like. And then you have a query pocket in gray. And what we do, we just simply overlay these two pockets, and we get the best overlay we can, and we get the score from this overlay, and we say, if your SiteHopper score is greater than 2, then your pockets are too similar, and we're going to discard them. And so we deduplicate based on the pocket similarity, not necessarily the protein similarity or the protein family similarity. In this way, we've curated about 1,900 protein ligand structures that are built into our data set that we've trained our ligandability model from. So from this, we've split it to about 1,500 training sets and about uh, 400 test data sets. And this is what it looks like. So we have a pocket surface patch representation, which is a positive control where you have a protein with a ligand bound. And based on a distance criteria, we draw a surface around this ligand, and we say that this area within this particular cutoff is a positive patch. And we have a negative control patch that I'm not going to talk about in glorious detail, but effectively it's a random part of the same protein that has a similar curvature and size as the positive patch. And that's what we're calling our negative control. And then we take this and we decorate the surface based on some distance criteria. And we have a feature vector that's constructed based on some point features that are taken from our toolkits that are based on some work from P2 rank. We're also inspired to use some atomic features that are projected back on the surface, again using our toolkits that was inspired by this GRASP work. And then we go ahead and we take all of our positive and negative control patches, we featurize them, and we train a model, either a random forest or a Gaussian process model. We found that random forest tends to be a little bit more performant, so the data that you see here will be from that, but they both are relatively well behaved. So this is how the model looks. We have a set of surface points similar to P2 rank, where each surface point has its own little bit of pocket score. And then we take the sum of the square of these scores and we call this our total score. And then we normalize this score by the number of surface points such that we can compare pockets of different sizes. So we don't want this score to just simply grow as the pocket itself grows, but rather we want to be able to define what is a ligandable pocket regardless of the size. And so this is how the data shakes out. In gray, you'll see our negative control points. In red, you'll see our known pockets from our data set. And you can see there is a separation that happens about 0.3 on our normalized ligandability score. Our AUC is quite high, so about 0.97. We think that this has a really reasonable way of separating positive from negative controls. And in order to find the actual breaking point, we use the so-called Bayes factor, where a Bayes factor is just the ratio of two likelihood probabilities. So the idea is that if you have the same data given a model, which is ligandable or not ligandable, how much evidence do you have that your model is predictive or not? And a higher Bayes factor, higher than 1, tells you that you're actually getting evidence for your model being predictive. We have adjusted this such that we have a Bayes factor of 3.2, and at this point, the ligandability score cutoff is about 0.27, so roughly about 0.3, as I was saying before when we were eyeballing it. So now it's time to look under the hood of our ligandability model, and I'd like to remind everyone of this really great quote from George Box that says, "All models are wrong, some models are useful." Now, we wanted to ask the question, are we biased towards pocket properties? Is that why we may not be, great in all cases? So here I'm going to show the normalized pocket score on the x-axis and the pocket similarity with the training set on the y-axis. And if we break it up with our known pockets in tan and our negative controls in gray, you can see that our known pockets have effectively no dependence on the pocket similarity with the training set. However, you can see that the pocket score actually slightly decreases as the score decreases, or as the score increases. And what that suggests is that we may have opportunities to pull out some of those pockets that may actually be real pockets that we are just assuming are surfaces that are negative controls, which actually may not be the case. So we'd expect this to kind of level out in the future, but this is what we see today. So the pocket properties that we're evaluating in this particular case are the buriedness, which is effectively the difference in your solvent accessible surface area and the bound state to the unbound state, and also the pocket polarity, which tells you about the roughly how much polar surface area you're getting relative to the total surface area. So if we look at the buriedness, what you see that indeed more buried pockets are scored higher. But that's not unsurprising because if you look at our training data, the training data also shows the same trend. So we're not really learning anything new here. We're just representing what is inside our training set. Also, if you look at pocket polarity, you can see it's roughly constant as a function of the normalized pocket score. And this is also a function of our training data. So again, we aren't learning anything, but rather we're seeing that we don't have this biasing our system and our ligandability model, at least for pocket polarity. So we do see that our ligandability model is slightly biased towards buried pockets, but that's because that's what our training set had. But the question is, how does it actually perform on known binding sites? And to answer this question, we just took out all of the known FDA binding pockets and applied our ligandability model to them. And what we see is that 81% of 121 drug binding pockets are predicted by our model to be ligandable, but you can see there are a few false negatives that fall below that 0.27 threshold. But if we look and we ask the question, what is actually contained in this set of false negatives, we find drugs that were stabilized by cofactors, big floppy things like peptides, peptidomimetics, macrocycles, covalently bound drugs with multiple occupancies, and so forth. Well, as a matter of fact, we purposely removed all instances of those from our training set. So we had no information about these. And so it's not surprising that we were not able to predict their ligandability, but this gives us yet another opportunity to improve our model in the future. And if we have more data for this, we can perhaps either fine tune our model for specific modalities or incorporate that in a larger data set and make a ligandability model that's tuned for all types of drug-like molecules. So we've tested our model on crystal structures, and these are really nice because they have, conformations that are completely stabilized by a ligand. But what about those structures that we predicted in the beginning of the talk from our MD simulations? If we use those structures, are we still able to predict ligandability in a reasonable way, even though we may not be perfectly forming these pockets? Well, if we take a look at our simulated cryptic pockets, what we find is that, again, the vast majority are shown to be ligandable, but the false negatives that we have are the same false negatives that we had with FDA-approved drugs. They're peptides, peptidomimetics. And we know these are missing from our training set, so we're not surprised to see these as our false negatives. But it gives us an idea about how we can improve the method for the future. Now, if we go back to our favorite system KRAS and we apply our ligandability model, the first rank pocket is the switch 2 pocket where all the FDA approved drugs have gone. The rank 2 pocket is the pocket on the other side of switch 2, the switch 1-2 pocket. So we think we have a method that is reliable and hopefully will shave off that 30-year wait for finding a pocket for one of the most deadly proteins in all of human cancers. So we know our biases and imperfections with our models. We know that these shallow pockets tend to get lower scores. That means maybe a pocket from a fragment screen perhaps will not get a high score. But we also know that the model is likely to fail for other types of binding sites, sites that are stabilized by a cofactor, sites with big floppy molecules in them, and sites where you have covalency involved. And we know that some of these are due to missing feature representations, but also we don't have a ton of examples for some of the big floppy things in covalent inhibitors in order to make a really strong model. So we're still looking into how to build up our data set for that. So again, what are the questions that we're asking here? Number one, we said, where might a ligand bind? And we tried to answer that by finding a method to push proteins around and detect cryptic pockets. We also asked the question, where does a drug-like molecule bind? And to answer that question, we developed a ligandability model that helps separate the false positives from the false negatives. And finally, what difference would a ligand make if it does bind to a protein? And this is a real question of biological efficacy, and we would love to have a partner to help solve this problem. We know this is bigger than just, a 1 or 2 month thing. So please talk to us if you'd like to answer that with us. So to conclude, I'd like to say that we've developed a method to extend the dynamics of proteins. And in that way, we can, kind of go back to Galileo and say, and yet it does move. And we've also found that Xenon is a pretty good probe for inducing pocket opening. And we've validated it on a large set of proteins. And we are currently writing up a manuscript that should be published later this year. And finally, we've also developed a ligandability model to help you rank these pockets so that you have a way to separate a good pocket from a bad pocket. And with that, I'd like to acknowledge all of those involved, primarily Neha and Jia and the Enhanced Sampling Group at OpenEye. Of course, this came from a collaboration with Black Diamond Therapeutics that lasted many years, and some suggestions from a good friend of ours, Dave Lawson, who was formerly at Mirati Therapeutics, and of course, the Westbrook community that makes this all possible. But all of this is already available in Orion to make your life easier, so go ahead, give it a try, let us know what you think. And with that, I am happy to take any questions that you may have, and we'll pass it over to Paul for moderation.
So, David, thank you. Magisterial as always. A few questions come in over the course of the talk. Warm you up with an easy one. How long does this take wall clock time to both do the pocket simulation itself and then to do the ligandability model prediction?
Yeah, that's a really great question. So originally when we started doing this, there's a lot of simulation that has to go into it. And so just the simulation time was taking about a week. Over the past few years, we've really tried to optimize the simulation part, and we've got it down to about a day and a half, depending on the protein size. Now, the ligandability model, since it is a model, it's much faster. But we have to, gather up all the data and analyze the full ensemble of our simulations. So that can take a few hours if you do it on your entire ensemble from weighted ensemble simulations. But if we're running it again on just a simple single system, it's really fast.
So if we were doing, say, predictions on AlphaFold or X-ray structures where you've only got one conformation, this could happen very quickly.
Very quickly, yeah.
So something a little more difficult now. I think something that came out as you were talking, that the special source of this is the Xenon in the simulation. So two things like that. First, why Xenon over any of the other noble gases?
Yeah, that's a good question. So what we know about noble gases is that if you look at the column that they fall into, all of the noble gases weirdly happen to be general anesthetics. And the efficacy of general anesthetics increases as you go down. And Xenon is the most efficacious that is also not radioactive. And so it has some really nice properties to it where it diffuses relatively quickly. It doesn't tend to aggregate like some things. And it can fit in nice hydrophobic little pockets. And so that's really why we like that as our part of our secret sauce.
So following up on the hydrophobic pocket idea, obviously there are lots of other solutions out there where we're using not noble gases, but hydrophobic fragments, again, benzene, isopropanol, and so on. Can you talk a little bit about why you pick a noble gas atom versus something like a hydrophobic fragment like benzene?
Yeah, that's a good question. So we picked it for a few reasons. Number one, we are only applying our enhanced sampling to the protein. So we needed a method that allowed really fast sampling of a probe-like molecule. And because Xenon has fast diffusion constant, even inside, proteins, we thought that we wouldn't need to enhance its sampling. We could just kind of get the protein to move around quite a bit. Another thing that we're really happy with is that this method is completely bias free. And so that means that we don't add any fictitious potentials or anything like that to keep like benzene from aggregating, which is a typical problem. And because of that, we're able to run our simulations in low concentration xenon. We don't get aggregation. We don't add any kind of bias to our system, which we don't actually know. And finally, in our KRAS study, we did try benzene out to see how well it would work. And as a matter of fact, it wasn't able to open up the switch 2 pocket. If you compared the reference pocket from the crystal structure to our observed pocket with either benzene or xenon, and you take the site hopper similarity, as it turns out, xenon was just much better at that. So we let the data kind of show us which direction to go.
The Xenon actually works well for some of those induced-fit pockets versus the conformational selection.
Yep, exactly.
I see. Okay. So this is a bit of a tough one. You talked a little bit just a moment about the time scale of the simulation and so on. And in some cases, pocket opening is known to take a very, very long time. How confident are we that we're able to simulate the kinds of timescales that we believe are involved in cryptic pocket formation?
Well, at this point, based on that large validation set that I showed, I think we're incredibly confident. So the types of motion that we can capture goes well beyond what you get from brute force simulation. So not just microseconds to milliseconds, but we can go well beyond that, seconds to minutes. So I'm fairly confident in that one.
Wow. So we can actually simulate it in seconds to minutes of actual physical time using this approach.
Yes, we can.
It's pretty amazing.
Yep.
Watch the space. Super. David, thank you very much as always.
Thanks, Paul.
Thank you very much for that. That's all we have time for today. So thank you all for attending. Thank you, Dave, for presenting. And we look forward to seeing all of you at the next webinar from OpenEye Cadence Biotech Sciences. Until then, goodbye.